The United States will need to conduct 193 million COVID-19 tests a month if we are to safely reopen schools, fortify COVID resistance in our nursing homes, and eventually see our way out of this pandemic. That's the conclusion in a report issued today by the Rockefeller Foundation and the Duke Margolis Center for Health Policy. Most of these monthly tests will focus on our schools, about 175,000 a month. But to reach these goals, the United States will need to greatly increase its testing capabilities. Right now, we're able to conduct about 30 million COVID-19 tests a month. Hi, I'm Frank Diamond, the managing editor of Infection Control Today, where we're trying to give infection preventionists and other healthcare professionals on the front lines the information they need to battle COVID-19. 193 million COVID-19 tests a month. A staggering number, but an achievable goal, according to the report. Many companies are developing all sorts of COVID-19 tests and trying to get them to market as fast as possible. As a matter of fact, coming up, I'll be talking to Oliver Schacht, the CEO of OpGen, one such company that's developing COVID tests. Mr. Schacht is going to give us a general overview of the importance of the sort of test development efforts being done by his company and a host of his competitors. He'll outline the challenges and promise of COVID testing. You don't want to miss my interview with Oliver Schacht that's coming right up. But let's pull back a little and get the big picture. In the United States, there have been over 6,300,000 confirmed cases of COVID-19. And in the United States, about 190,000 people have died from COVID-19. In the world, there have been nearly 28 million confirmed cases of COVID-19. And in the world, nearly 900,000 people have died from COVID-19. These statistics brought to you by Johns Hopkins University. Now, back to that report by the Rockefeller Foundation and the Duke Margolis Center for Health Policy. It's gotten a lot of attention today, as you might expect. The authors state that the tests don't necessarily have to be the most accurate kind of test used to detect COVID. Screening tests that are less accurate but that can deliver results faster should suffice in many instances. This news about testing comes at a time when health experts brace for a second wave of COVID-19. But while some health experts predict a spike in COVID cases, they also say that fewer people will die from it. The ratio of death to cases will be smaller than in the first wave. That's because the medical system has gotten much better at treating people with severe symptoms, and older people and people with comorbidities are more careful when it comes to social distancing. That said, more people will die from COVID, perhaps 224,000 more people by the beginning of next year. That's according to a COVID tracking model created by the Institute for Health Metrics and Evaluation at the University of Washington. It predicts that over 410,000 Americans will die from COVID-19 by January 1st. That's a lot to digest, and we're trying our best here at Infection Control Today to give you, infection preventionists and other healthcare professionals, the information you need in a digestible form. Please go to our website at infectioncontroltoday.com. Hi, I'm Frank Diamond, the managing editor of Infection Control Today, where we hope to give infection preventionists and other healthcare providers on the front lines the information they need to battle COVID-19. But as infection preventionists know all too well, there are still other antimicrobial resistant pathogens out there. They didn't go on vacation when COVID arrived. COVID-19, as you know, is caused by a virus, SARS-CoV-2. The pathogens that have been given hospital infection prevention fits for decades now are, for the most part, bacteria that have grown resistant to antibiotics. The superbugs, remember them? And a lot of times, hospitals have to, at first, give patients with a bacterial infection a broad spectrum of antibiotics until diagnostic testing helps them pinpoint exactly what they're up against. That can take precious time. What's needed is faster diagnostic testing. That brings me to my guest, Oliver Schacht, the CEO of OpGen, a company that bills itself as harnessing the power of molecular diagnostics and bioinformatics to help get better information to healthcare providers faster. Mr. Schacht is going to give us a general overview of how important these sort of efforts by OpGen and other companies his competitors are. Mr. Schacht, thanks for being with us today.
Pleasure. Thanks for having me.
When I first started covering this beat back in November, antimicrobial resistant pathogens was one of the main concerns for many of my readers and others in healthcare. Then COVID came along and of course sucked all the air out of the room. But this is still a big problem, isn't it?
Absolutely. And you know, many folks these days come to realize that it's really the pandemic underlying the current COVID-19 pandemic. As you rightly said, you know, the science has been around for 20 plus years on antimicrobial resistant infections. And it's a bit like climate change. It happens at a gradual pace, but it's going in one direction and one direction only. It's getting worse. Bugs are becoming ever more resistant. Even when new antibiotics, as scarce as they are, come to market, they quickly lead to more resistance by certain pathogens being adopted. So really, in the absence of many new antibiotics, because a lot of big pharma have also gotten out of the development of new antibiotics, leaving it to, frankly, small biotechs. And even those portfolios and pipelines are fairly dry. So the only weapon we have at our disposal to maintain antibiotics as an effective weapon against these superbugs is rapid diagnostic testing. As much as we're learning about the importance of rapid testing in COVID, I think it's the same name of the game, rapid testing for bacterial superbugs in hospitals. And that's really where the company, as OpGen, as well as our recently acquired and merged entity Curatus in Europe, have been a part of for over a decade, developing novel rapid diagnostic solutions for detecting not only the superbugs, but also their antimicrobial resistance.
And you said that the problem keeps getting worse. How so?
Well, if you look at the percentage rates of what percentage of certain pathogens have already become resistant across many different geographies and across the different types of bacteria, what we're finding is that for many of these bugs, 15, 20 years ago, it was all about MRSA, you know, gram-positive, Staph aureus, getting resistant to methicillin. That was sort of the big scare, MRSA. A lot of testing, a lot of rapid testing was developed. Rates have actually been going down. And on the flip side, a lot of gram-negative bacteria have recently gotten their claim to fame. And in some hospitals and some bugs, we see resistant rates in the 30 to 40% rate, i.e. every third, sometimes, you know, almost approaching every second bug are already resistant to one or more antibiotics. And as some recent high-profile cases have shown, there are bugs that are by now resistant to all but, you know, just the last resort antibiotic or even all antibiotics, leaving us with absolutely no options. So finding the bug and detecting its resistance profile has become that much more critical given these rates.
As you know, my readers, many of them are infection preventionists at hospitals. And what you are talking about seems to be part of the antibiotic stewardship teams. And sometimes infection preventionists are on those teams or take part in some way, and sometimes they don't. What's your feeling about that?
There's no doubt that antibiotic stewardship is a key component. And in fact, it's getting its fair attention these days. And a lot of hospitals are waking up to the fact that it is without improved antibiotic stewardship, and this really being an interdisciplinary approach between the clinicians, the microbiology lab, the pharmacy, and you know, sort of the infection prevention folks. It's got to be an interdisciplinary, multidisciplinary approach on a patient-by-patient case in order to get the best possible treatment. It's really about personalizing antibiotic therapy for an individual patient based on the individual bug and its resistance profile. Because in the absence, go ahead.
But where do you see infection prevention fitting in here?
Well, it's really making sure that interdisciplinary communication happens. It's always easy. I mean, you're a clinician on an intensive care unit. You're worried about treating an individual patient who may be fighting for their lives. If you're in the microbiology lab, you know, all sorts of clinical units send specimen. You're worried about your budget. You're worried about the throughput and the technician time it takes. You're in pharmacy. You want to make sure, obviously, the best possible treatment gets used, but also the most economical, and really bringing all those different priorities and disciplines together and making sure that, you know, you treat as broadly as necessary, i.e. use broad spectrum wherever that truly indicated, but as narrowly as absolutely possible to make sure that you do not consistently overtreat. And we've seen this, you know, on plenty of published data sets where, you know, on a cohort of 600 plus patients in Northwestern and Chicago and Beaumont, Michigan, they've done chart reviews and basically shown that more than half of all patients were inadequately treated, overtreated with broad spectrum antibiotics. So if you know that you're not facing a superbug situation, you may as well start thinking about de-escalating and taking away some of that broad spectrum coverage to really preserve that for those cases where it's warranted. And on the flip side, if you do know you have a certain superbug, you may want to add a certain specific therapy to make sure you're really addressing that particular bug and that particular resistance profile.
Does the diagnostic testing start in the emergency department?
It could be in the emergency department. So sometimes, yes, if you're thinking about sort of entry triaging, but we're also seeing this with patients who are already hospitalized, who are already inpatients. They may be hospitalized for something entirely unrelated. They might be cancer patients or cardiovascular patients, transplant patients, burn unit patients, and many of them get the infection in the hospital as a secondary disease, if you will. And if they're already hospitalized and they're already on, you know, one of the many potential clinical wards or intensive care units, and so even for those patients, you need to make sure that you get that rapid testing. You take the specimen, you're sending it to the lab. They're invariably going to run microbiology culture, which, you know, again, hasn't fundamentally changed in over 130 years. It's been automated somewhat, but it's still a good, you know, 2 days, sometimes 3 days and more to get the full picture on the bugs and antibiotic resistance or susceptibility. So having the ability to use the same specimen, a small portion thereof, and run a quick molecular test in the same lab where you get an answer after 4 to 5 hours is huge, because it does allow clinicians, in collaboration with stewardship committees, with the lab, to reassess whether or not their initial selection of broad spectrum antibiotic is appropriate or not.
Who does this well? I mean, if you want to name names, that's great. Hospital systems in the United States. You've been around, you must have seen how some hospitals do it. Who does it well?
I don't think anybody has found sort of the silver bullet here yet. I think there is a growing awareness, and clearly, if you look at, you know, a lot of the adoption rates for some of these fast systems, you know, like the BioFire system from bioMérieux, QIAGEN systems, QIAstat, the Univero system from Curatus and OpGen, GenMark systems. So you do see a general adoption. It's started out going fairly well in upper respiratory tract infections, mostly viral, a few bacteria. It's also going reasonably well in the case where you already have a blood culture, because a lot of blood culture automation has happened, and so there is a high throughput there. Where clearly there is a scarcity of testing to begin with is areas such as lower respiratory tract infections or pneumonia, mostly bacterial. And in fact, we're seeing this in the context of COVID. There's a significant proportion, depending on the study, somewhere between 25 and 40% of COVID patients end up also getting a pneumonia. It's also still fairly underdeveloped in areas such as sepsis, straight from whole blood, or even things like prosthetic joint infections. You think about hip and knee replacement, we're still waiting for culture results for 10 to 14 days. With half of those results coming back negative, you're none the wiser. So I think while we've gotten pretty good in certain areas, there's still a lot of disease states or disease areas, such as pneumonia, such as also complicated urinary tract infection and orthopedic joint infections, where these systems have yet to really make a big impact in medical practice.
Has the CDC or any other government agency pointed out that this is such a problem, and have they tried to remedy it in any way?
Well, you look at the realities again. The World Health Organization published a report 20 years ago, the year 2000 or 2001, called Bad Bugs, No Drugs. That report, was sort of updated in 2018, I believe it was, not only confirming that all the predictions, dire as they were, had become true. In fact, it was a lot worse. In fact, resistance rates are higher. Resistance was adopted or acquired by bugs faster under new antibiotics than anybody had anticipated, much like climate change. Again, 20 years ago, we knew there was climate change. It wasn't until these last 2-3 years that we've really, as societies, started making dramatic wholesale changes, whether it's our energy generation, whether it's automotive or mobility. And I think, again, we've got to see the same thing. So while entities such as the World Health Organization, the G8 and the G20 have had antimicrobial resistance as one of their key themes here in the last couple of years. So there's no lack of political attention or political will. But when it then boils down to implementing it into medical practice, you've got to realize that this is a global challenge. It's not something you can address at a local, regional, not even a national level, because we travel, at least we used to before COVID, these bugs spread so quickly that doing something in just one part of the world, one country, is meaningless unless everybody is really doing it. So this really requires a global, supranational approach. And again, the attention is there. We now got to make sure there is appropriate funding in the medical systems, not only for high-end drugs, but also for rapid diagnostic testing. And that, I think, is one of the areas that certainly the whole diagnostics industry has struggled with, frankly, is also struggling with in COVID right now, is consistency, predictability when it comes to reimbursement of rapid molecular testing. Because only when you can plan and you have a certain degree of assurance that once you've developed these types of tests, they're also going to get reimbursed at a level that make economic sense for companies to do so, will they get developed and will they be made available widespread.
Where will the funding come from? Does it have to be government-based, you think, at this point?
I mean, there's got to be certain government incentives. I think when you start looking at certain healthcare systems that are better than others, when you start looking at it holistically, if you only look at your lab budget, any diagnostic test that you're adding into the system will generate net incremental expense. The saving, on the other hand, whether it's saving reserve antibiotics or saving the usage of broad spectrum antibiotics, and ultimately the saving of lives and saving time spent on intensive care units, those savings will not be realized by the lab. They'll be realized by either the clinical ward or ICU, or even the hospital system at large. It may not even be visible to either the lab or the clinician. So unless you take that global and holistic approach, I mean, if you're looking at, say a moderately sized hospital with 1,000 pneumonia cases a year, I mean, you're looking at 1,000 tests that could run, anywhere in the $150 ballpark per test. You've got to find that incremental budget of $150,000. If you then look at how many lives it could potentially change and save, and how many treatment courses might be spared, I think there's a good case to be made for this more than paying for itself. But again, you've got to take that holistic approach and look at it not only from a local lab budget perspective.
Is there anything pertinent about this that I neglected to ask you, especially as it pertains to infection prevention at hospitals?
Well, we've seen some great initiatives. For example, the New York State Department of Health has partnered up with OpGen about a year and a half ago, and we've developed with them, together with the Wadsworth Center and 3 large New York City health networks with roughly 3 dozen hospitals, what we call the Lighthouse. So this is really a tracing and tracking bioinformatics solution that allows identifying rapidly and then tracing patients who may have had a multi-drug resistant bug, who may, for one reason or another, have been transferred from one hospital or one unit in a hospital to another one, thus spreading potentially these superbugs. And again, having that ability to rapidly test, but then also rapidly share the information from a bioinformatics knowledge-based standpoint has been tremendously helpful. And this has been in collaboration not only with the New York State Department of Health, but previously Merck Illum, more recently a company called IDC, after they acquired Illum from Merck. And again, it's a multidisciplinary approach that has rapid diagnostic testing, has the hospitals, clinicians, and labs involved, and also has this bioinformatics knowledge-based component that really allows states across multiple healthcare systems to trace and track and hopefully help prevent these types of super infections.
Oliver Schacht, thank you so much for visiting here at Infection Control today.
Been a pleasure. Thank you.